Hi, I'm Mr. Williamson, and this video is about what happens to the NADH2s and the FADs from glycolysis in the Krebs cycle, or the electron transport chain. Before we get started, I would suggest taking good notes. Outline, Cornell style, you're the boss, applesauce. Hey, do it any way you want, but take notes because it helps you. Just the process of writing the notes down helps you learn. Okay? If you have any questions, make sure you ask in Edpuzzle or in the classroom. I'll be glad to answer them. Let's get started. So the question, as I said, was what happens to those NADH2s and the FADs that were split off every time where a hydrogen ion was removed from the glucose. I said it was stored in NAD, becoming NADH2. They're just holders. All they do is hold the hydrogen. They store it for a little while. Well, anyways, those move out to the membrane of the mitochondria. So where we used to have the mitochondria, the Krebs cycle, which took place inside the inner membrane, we're now moving outside to the outer membrane, to the membrane of the outside of the mitochondria, which is where the electron transport chain takes place. Basically, electron transport chain is a series of oxidation reduction reactions that move an electron down an energy gradient. All right. Think of the energy gradient kind of like diffusion from a high concentration to a low concentration, and that's what this is. We're moving an electron from a high energy concentration to a low energy concentration, and we need a place to put that, and we put it on oxygen. So it works kind of like this. This is a bucket brigade at a fireman's muster. What they have to do is fill that barrel that's at the bottom of that shack or that shed. It has a rain spout that goes into it, and they fill it up bucket by bucket. So what we're looking at here is waiting for the right moment. Just one moment here. Okay. So what we're looking at is this water that is spilling out in this area right here. All right. Think of that water that's spilling out of the bucket as energy that is being released from the electron, from the electron transport chain that the enzymes can use to attach a phosphate to ADP to make it ATP. So every time the bucket goes from person to person to person, it spills out some of this water. All right. And that spilling of the water spills out some of that energy. That spilling of the energy is what allows it. The energy that is allowed to be used to attach a phosphate to ADP to make it ATP. The problem is that if you notice as I start this again, but as we go, the bucket, this bucket right here is going to be passed to this person, who's going to be passed to this person, who empties it up on the shed and then hands it back to this guy. The problem is, what happens if this guy here doesn't take the bucket? The whole thing stops. All right? And that's what oxygen does. Oxygen allows that electron to move down the electron transport chain steps, spilling out energy along the way, and then oxygen comes in and picks it up, and it allows it and takes it off, goes. And when that happens, that allows the next one to come along and do the same thing and to come attach to oxygen and go away, which allows the next one to come along, and every time spilling out a little energy, allowing the phosphate to be attached to ADP to become ATP. The problem is that if this doesn't happen, if there's no oxygen, this last electron doesn't go anywhere. So this one can't start. The electron transport chain stops. Oxygen is the final ion acceptor in the electron transport chain that allows it to keep going. Just like this gentleman right here has to take the bucket from this person to hand it to that person. If this guy doesn't take the bucket, then the bucket just stays over this side right here, and he can't do anything. The whole thing stops. That's what oxygen does. It allows the electron transport chain to keep functioning. The final ion acceptor is oxygen. It's accepting the hydrogen ions. So if you have oxygen and we add, let's call it 2 hydrogens, what do we end up with? Water. Oxygen is the final ion acceptor. It accepts 2 hydrogen ions to be water. So you're wondering what happens to the oxygen after you breathe in. It doesn't become carbon dioxide. It actually becomes water. Without the oxygen, those hydrogen ions stop up the electron transport chain and it no longer works. As I said, the energy generated by the movement, that spilling of the water, is used to make by the enzymes to form ATP, to attach a phosphate with the energy in that high energy bond between the third and the second phosphate to make ATP. Now, the way it works, for every NADH molecule, we're able to make 3 ATPs. For every FADH molecule, we can make 2. So we got to figure out how many NADH2s do we make and how many FADs do we make. Well, there were 10 NADH molecules and 2 FADs, and I'll show you where we got them each. First of all, remember the glycolysis? And this will happen eventually. There were these 2 here. These 2 NADs. So that's 2 NADs right now. 2 NADH2s. All right. We had an NADH2 here. That's 3. NADH2 there. That's 4. NADH2 there. That's 5. And this one was an FAD. So that's 1 FADH. And then we go back to an NADH2 here, that's 6. So far that's 6. So how did we get 10? Remember, there's 2 pyruvic acids per glucose. So we do it again. This is 7. This is 8. This is 9. Here comes the second FAD. And then finally, this is the 10th NAD. So 10 NADH2s, 2 FADs. Like I said, 10 NADH2s, 2 FADHs. So a total of 34. 10 times 3 is 30. 2 times 2 is 4. 34. All right. Glycolysis cost us 2 ATP to form pGal. Remember, we had to use a little bit of ATP to get this whole thing going, to get to split the glucose molecule into pGal. But then afterwards, when we split up the hydrogens, we actually got made 4, so that's a profit of 2. All right, so this one cost us 2, and here we get a profit of 4, or got a plus 4, and that's a profit of plus 2. So we're up 2 so far just in glycolysis. We add another 2 of them in the Krebs cycle. Right here. All right, so that's 1, but remember, we go through it twice, so there's 2. So that's another 2, so that's 4. So we start out with 34 from the electron transport chain, 2 from glycolysis, and 2 from the Krebs cycle, 38 ATP. So for each glucose molecule that goes into the system, goes into your cells, and it starts glycolysis, we get 38 ATP. All right. Now, sometimes in some textbooks or online, you might see 36, and in reality, it actually is usually less than that because the system is not totally efficient. Sometimes parts of it break down, the enzymes don't work real well. But in this class, for testing question purposes, we are going to go with 38. How many ATP do we get from glucose? We get 38. That's the bottom line. All right. So now we can actually answer the question, why do you need food and oxygen? Before it was just because you'll die. Yeah, but why? You don't know. Some of you knew it had something to do with energy, but now we can be really specific. We need food for glucose. Glucose contains the bonds, the energy in the bonds, in a form that we can break those bonds in glucose and create energy enough energy to make ATP, a form of energy that our cells can use. All right. So why do we need oxygen? Oxygen is the final ion acceptor in the electron transport chain. Without oxygen, we don't get 34 ATP out of the 38. We only get 4 out of the 38. Remember, 34 is in the electron transport chain, 38 total. That only makes 4. That's not very efficient. That's not enough to sustain us. We can't keep living that way. All right. So food supplies glucose. Oxygen is the final, and you need to know this, oxygen is the final ion acceptor in the electron transport chain. So in summary, sum this whole thing up, the last 3-4 videos. We need food for glucose. Glucose contains energy in the bonds that our cells can use and break down and change into the energy that, remember, because you cannot create or destroy energy, and change into energy that could be in a form of ATP. All right. The process, basically, glycolysis turns glucose into pyruvic acid, where we get 2 ATP. Pyruvic acid moves into the inner membrane of the mitochondria for the Krebs cycle, which basically is making NADH2s and FADHs, and we also get 2 more ATP. Those NADH2s and FADHs go to the electron transport chain with oxygen to make the 34 ATP to give us a total of 38. Without the oxygen, we only get the 2 here, or 2 each there, 4. Oxygen is what allows us, the electron transport chain, to get rid of that electron that moves down that electron gradient, to move down the electron gradient, and oxygen comes along and takes it away, so the next one can move down the electron transport chain and then be taken away, and the next one can move down the electron, and so on and so on. We stop that. If the electron doesn't move, it jams up the entire electron transport chain. It no longer works. Oxygen is what allows us to get these 34 ATP. So one more time, let me summarize it. We eat because we need glucose. Glucose contains energy in its bonds that we can break and change into energy to make ATP that can be stored in ATP. It starts in glycolysis, which takes place in the cytoplasm. Basically, it costs us 2 ATP to do that, to break into pyruvic acid, or PGAL, don't worry about that, but into 2 molecules, which becomes pyruvic acid. But we get 4, so a profit of 2, plus 2 NADH2s. Pyruvic acid moves into the inner membrane of the mitochondria, goes through the Krebs cycle twice because there's 2 molecules per glucose. We get 2 ATP, we get 10 NADH2s and 2 FADHs, which go to the electron transport chain in the membrane of the mitochondria, giving us 34 more, so we're a total of 38. Hopefully, you understand it. If you don't, please ask me questions. I want to make sure you understand that. That's the overall picture. I know all those details on those videos before, and this one maybe even, you're just like, oh my God, I got to remember it. No, you don't. Be able to summarize it, just like I just did. All right? Hopefully you took good notes. Hopefully if you have questions, you ask them, you let me know. Good luck.